गुड मॉर्निंग मैडम। गावंडे सर
Now today, our topic of discussion is of genome evolution in timeline. Because I want to share two parts. One is of from the evolutionary genomics, and other from the amino acid evolution in timeline. Because whenever there is evolution, whatever the things are there during the primordial soup, because we tie up our evolution right back 3.5 billion years ago. Once there is a formation of primordial soup, and ultimately there is a formation of first living organism as in the form of prokaryotic cell. Then thereafter there is a multicellularity, eukaryotic organization, and then multicellularity, and ultimately today we have this beautiful world because of the evolution. When we start reproducing, when we start accumulating all the cells, and ultimately there is a formation of organ and then system, and ultimately there is a formation of organism. But prior to that, one most important thing is whatever evolutionary changes during this period, that is because of the continental drift. One most popular book written by Alfred Wegener, The Origin of Continents and Oceans. Now, he talk about the creation of supercontinent first. Only one ocean is there, right? 160 billion years back. Only one ocean is there. That ocean, we popularly called it as a Panthalassa. Now, two continents are there during that period, Laurasia and Gondwana land. Now, gradually there is a shifting, and ultimately this beautiful world come into picture. That is because of the continental drift. Now, this supercontinent are formed around 335 billion years ago, and ultimately there is a continental drift. During Permian period, the Pangaea is a continent. Now, due to the tectonic plate shifting, during Triassic 200 million years ago, there is a Laurasia and Gondwana land. Now, gradually, Jurassic to the Cretaceous period, then Cretaceous period again divides into two, that is Upper Cretaceous and Lower Cretaceous. And ultimately, here is this beautiful world that get it separated by the virtue of continental drift. Now today, these are the continents that we seen on the world map. Now, the eventually evidences that put forth by Alfred Wegener, that the evidences are there. One of the popular evidences is of the matching of continents, that he called it as a zigzag soffit, rock of the same age across the oceans, tillites, lesser deposits, and distribution of fossils. These evidences that create advocacy towards the separation of this globe. Because of, here you will find in Africa and South America. This is what the zigzag saw-like structure. The faces get it matched. There is matching of faces are there. Then here is the Eurasia, then to the North America. When we put all this together, there is a creation of a super continent. Now there is a separation of continents. Whatever tremendous environmental pressure is there, and because of this tremendous environmental pressure that intended to get it separated, and ultimately there is a formation of the today's a beautiful globe because of the separation of oceans, because of the separation of, because of the shifting of these plates that we called it as a tectonic plate shifting. Now here is a, when we're talking about the matching of the zigzag swords, now, and the folded mountains, because Himalaya is a folded mountain, the matching of folded mountain ranges that related to the deposition of materials and minerals. That's why this assumption is of, that means there is a only one supercontinent is there. After the tectonic plate shifting, there is a shifting of, during that time, there is a shifting of these continents, and ultimately there is a separation. Now, when we're talking about the tilites, you will find there is a matching of tilites. That's why whatever the case is there, that we always consider the Pangea is a supercontinent, and ultimately get it separated. That is because of the similar type of deposition as in the permaphilites. The placer deposits, coal deposits, there is a belt. This belt is of the coal belt, and now the coal deposition belt. If you analyze the coal deposition belt, then you will find there is a creation of this this different type of continents because of the tectonic plate shifting. Now, another thing is what the distribution of fossils. When we consider the deposition of fossils, there are many more evidences by fossil deposits that created this beautiful world, beautiful universe, or beautiful world by the virtue of tectonic plate shifting. This is what another thing. When we consider the evolutionary trend, then you will find during the tectonic plate shifting, because the Arctic and Antarctic. Arctic is the ocean, and the Antarctica is a continent. That's why the only one place on this globe that need not require any visa, right? Immigration and immigration authorities are not there, because we started our journey from the Antarctica towards Arctic. Now, earlier the condition of this globe is, that is what the Big Bang theory and black hole theory that states. Earlier this globe is too much warm, too much hot, sorry. Thereafter there is warming. Later on, the formation of small droplets of water because of the condensation of ammonia. Now after the formation of droplets, now the situation is again win-win situation, and ultimately there is a formation of a semi-solid structures, semi-solid material that we called it as a primordial soup, wherein the primordia of cell that get it arose. Now, there are millions of amino acids and nucleic acids that get it accumulated inside the primordial soup. Now, only the way of the accumulation of these amino acids and nucleic acids, that is because of the nitrogenous. Nitrogen is a material available in abundance. Thereafter, there is a carbon. Now, the carbon accumulation, because of the tremendous environmental pressure, there is accumulation and conversion of this carbon into the amino acids and nucleic acids. Now after the formation of millions of amino acids and nucleic acids, now the chain reaction that get it started inside the primordial cell, wherein there is a formation of polymeric chain of these nucleic acids first. Thereafter, as on the basis of a electromotive force, as on the basis of adhesive force, as on the basis of the ionic bonding, there is accumulation of these amino acids around the nucleic acid, wherein there is a creation of the first cell that we called it as either we called it as a protist or we called it as a monomer. Thereafter, that whatever the things are there, that means the beginning of all these things, that is from the very tiny particles, the small particles. Now the small particles get it polymerized, not by the virtue of the biochemical processing, but by the virtue of whatever the physical forces are there at the time of primordial soup formation. Now, whatever UV radiations are there and that the ultraviolet radiations, ultrasonic sounds and all other things that tremendously that creates these biomolecules first. Now, whatever entity get it created inside the primordial soup, that we called it as a first cell. Now, whatever the things are there, that things come into interaction. There is a creation of the biochemical reactions, that whatever the forces are there, that whatever kinetic energy that molecule have, that kinetic energy get it again utilized for the creation of molecules. That is, whatever potential is there, that potential get it converted into kinetic, and kinetic energy is able to create the working biomolecules. This is what the thing. But the question is whether there is a formation of protein first or nucleic acid. That means when we're talking about the formation of this beautiful living world, that cell is an entity. Prior to eukaryotes, the entity is prokaryotes. But what happens when did the prokaryotic cell emerge? What happens at that time is what the too many the logical questions are there, because we are not at all part and parcel at that time. We cannot have the too much history of human civilization. Whatever the things are there, we predict that things as on the basis of whatever the paleobotanical evidences are there or paleobiological evidences are there. Now, the question is whether there is a formation of the amino acid first on this planet, on the primitive Earth, or the nucleic acid first. Now, the answer to that question is what? Because of the nitrogen, the availability of more nitrogen, if you taken into consideration of your cell, the very first entity come into picture is of the nitrogen. The material is nitrogenous material. Thereafter, there is a carbon. And now, the carbon accumulation because of the creation of many more biomolecules by the virtue of carbon. But prior to that, there is a nitrogen. Now, when we're talking about the amino acids, then you will find the amino acids that fall under the category. That means the categories are there under the amino acids. One is the positively charged, negatively charged, then hydrophobic, hydrophilic, neutral, as well as there is mixing. The aromatic and non-aromatic amino acids. Now, when we consider the aliphatic hydrocarbons, because after that there is accumulation of carbon. That's why the first biomolecules come on this planet that come into picture. That is because of the formation of aliphatic chains, small chain molecules. Thereafter, there is a creation of the aromatic hydrocarbons, the aromatic compounds, the aromatic amino acids. That means the beginning of or the reaction during the primordial soup is that started with a creation of simple molecules, as in the form of aliphatic hydrocarbons. Thereafter, there is a creation of aromatic hydrocarbon. Now there is amalgamation of aromatic and aliphatic hydrocarbons. And ultimately, there is a formation of the aliphatic amino acids and aromatic amino acids. Now, there are some experimental evidences for the creation of amino acid first, prior to the creation of the genetic material, as in the form of maybe as in the form of DNA or RNA. But the first material on this planet, as in the form of genomic material, that always in the form of RNA. Okay, later on I will talk on the whether how there is RNA stability, how the RNA get converted into DNA. But prior to that, the very first material come into picture is of the amino acids. Now, during the prebiotic synthesis experiments, most efficiently produced amino acids are the glycine, alanine, valine, and aspartame. You know, the evolutionary experiment that designed by Miller. And now Miller find the most abundance and efficiently produced amino acids are glycine, alanine, valine, and aspartame. These amino acids come into picture first. Thereafter, there is a creation of the aspartame. The most stable and the achievement of stability that always tie up with the aspartame, because aspartame is negatively charged amino acid and is unstable chemically. The another thing is what? The metal ions solve two problems at once. Right? Because whenever there is a reaction, reaction that always needs to require some sort of chelators. The metal ions plays very important role in order to create chelating potential. Now this chelating potential able to drag the charge towards amino acid that becomes positively charged, and that's why there is a stability. But being a biologist, we know the initiation codon that always starts with a methionine. Whether methionine comes first, whether methionine is a part and parcel of the primary polypeptide, whether the methionine evolved first, the answer is no. At what instances methionine is an amino acid, that amino acid treated as a initiation amino acid. Whether the initiation codon that always starts with AUG, the answer is no. Because during this experimentation, during the experimentation that the protocol developed by Miller, he able to find out only 5 amino acids that produced. Now recently, and in the recent past, the Higgs boson experimentation is there, and Peter Higgs awarded Nobel Prize for Higgs boson particle experiment. They create protocol for the sake of the creation of this universe. Now, during the creation of this universe, these two small particles are there that we called it as a protino and neutrino. Now this protino and neutrino particle get it fused. And because of the fusion and fission reaction, there is a huge explosion. And because of the explosion, there is a creation of a something balloon-like structure, gaseous balloon-like structure, hot gaseous balloon-like structure. And ultimately, during the explosion, there is a creation of the universe. Many more galaxies are there. We are the part and parcel of Milky Way galaxy. And now, a small piece on the universe is of this Earth. Now, during this explosion, this helium and the other particles that get it accumulated. During this accumulation, there is a tremendous nitrogen that get it accumulated. That's why there is a creation of the amino acids by the accumulation of whatever nitrogen is available. Now, obviously, there is a creation of amino acids. Prior to the creation of billions of amino acids, only 5 amino acids get it synthesized. Methionine not at all a part and parcel of the amino acid that come into picture during the primordial soup. There is a stability of amino acid that achieved because of these metal ions. That is again, there is a creation of positively charged, negatively charged, hydrophobic and hydrophilic amino acids. Now, the prebiotic amino acids that noted by Miller in 1955, a similar case by Johnson in 2008, and Ring in 1972. The reduction mixture of this CH4, NH3, that is ammonia, H2 and H2, that produce only the glycine, alanine, aspartame, glutamate and serine, and significant amount. During that experiment, valine was produced as similar to the glutamate. The order of abundance is of alanine, glycine, aspartame, valine, leucine, serine, isoleucine, proline, and proline. But the earlier there is alanine. But nowadays, only methionine is amino acid, is a starting amino acid that get it accumulated in each and every polypeptide. But prebiotic experiments that shows the order of abundance, exhibits the order of abundance as in the form of alanine, glycine, aspartame, valine, leucine, this much amino acids are there. Now today we tie up our findings to the occurrence of 20 naturally occurring amino acids and another is of the biosynthetic amino acids. Out of these 20, 13 amino acids that able to do the job for the formation of polypeptide. Now, when we talking about these amino acids, another one is the cysteine that was found in photolysis of this material during Miller's experiment. Now, another thing is what? The pyrolysis of hydrocarbons. The pyrolysis that creates phenylalanine, tyrosine, and tryptophan. Because earlier there is alanine, later on there is a phenylalanine. Tyrosine as a biosynthetic and complete entity, and tryptophan that get it introduced after the creation of eukaryotic cell. Conversion of prokaryotes to eukaryotes. Later on, tryptophan. Again, tryptophan is a biosynthetic amino acid. Later on, this tryptophan is the most important, not a bender, but most important amino acid that needs to require inside the cell in order to perform a different biochemical functions. Now let us consider the RNA word, because you know the RNA is the most important thing that created in the primordial soup. Inside the primordial soup, there is a creation of the nucleotide. But bases, millions of bases are there. But more than 10 bases that able to stabilize. Out of which 5 bases that these bases we called it as a total stabilized bases. Adenine, guanine, cytosine, and thymine. Adenine, guanine, cytosine, and uracil. That means the thymine, uracil, adenine, guanine, cytosine, thymine, uracil. These 5 bases. These bases, stabilized bases. Later on, there is modification of bases. Many more modifications that get it added. And created the modified bases. And these bases we call it as stable bases. Now, mandatory requirement for the creation of cell is of the one thing is amino acid. Eventually, that get it converted into polypeptides by the virtue of peptidyl linkage. Another thing is what? The message. That message written on the RNA, not in the form of DNA. Now, whatever message is there, that message get it identified by the amino acid. Thereafter, there is a formation of polypeptide. There is a strong evidence of this, the RNA word, the creation of first genetic material as in the form of RNA, not as in the form of DNA. That's why we're talking about the RNA is the first genetic material come into picture on this planet. Now, later on, there is a polymerization. Now, the creation of the larger RNA. Earlier, there is a very synchronized and rhythmic manner. There is the appearance of the RNA. Now, whatever nucleotides are there that identified by the virtue of the codes, the virtue of the ionic interaction. Now, this ionic interaction drag amino acids on proper position Thereafter, there is a polymerization, and ultimately there is an encapsulation of RNA inside the protein envelope. Now here is a creation of the environment. That environment is able to carry out the many more biochemical reactions. And ultimately, there is a creation of cell. Now, prebiotic synthesis of deoxyribose from glyceraldehyde and acetaldehyde, that is aldehyde, the aldol condensation reaction from glyceraldehyde and acetaldehyde is poor. That's why the aldol condensation reaction not at all work during the prebiotic environment. However, there are many more evidences for the creation of ribose. That is not the better combination. Because deoxyribose as during aldol condensation is the response is poor. Not at all. The ribose synthesis that didn't have the better experience for its creation. That is ribose 2. That is the glyceraldehyde and acetaldehyde, that is aldol condensation. Not at all as in favor to the creation of deoxyribose, as well as the prebiotic synthesis, not at all have to have the very conducive environment for the creation of ribose. But still, we create advocacy towards the RNA as a genetic material, not at all the DNA as a genetic. That's why the thing is there. But every evolutionary trend that ended with complexity. At the beginning, there is a simple molecules. The simplicity is there. That simplicity leads to the RNA. Now, there is a strong argument in favor of the creation of DNA before protein synthesis. Now, one thing is what? The transcription. Transcription is simple as compared to translation. Because, the RNA polymerase need not require any 3' hydroxyl group to its polymerization. But when you're talking about the polymerization in DNA, by the virtue of DNA polymerase, that needs to require 3' hydroxyl group. Because no primer needs to require for RNA polymerase. No 3' hydroxyl group. Subsequently, only the simple ionic interaction that leads to the formation of a smaller piece of RNA. But when you're talking about DNA polymerization, the 3' hydroxyl group that always needs to require for polymerization. But the situation is win-win situation that created intermittently. That is from RNA, there is a creation of DNA. That too single-stranded DNA. Thereafter, there is a double-stranded DNA. This task overcome by RNA dependent DNA polymerization, and that we called it as the reverse transcriptase. Because of the reverse transcription, there is a creation of DNA, no doubt. But very simple, very true. For the creation of RNA. The creation of RNA is of, that is what? The creation of RNA, that is because of the simple, there is a creation of RNA polymerase. Now this RNA polymerase is able to do the job without 3' hydroxyl group. And ultimately, there is a creation of a short stretch of RNA. Now this short stretch of RNA that able to do the job for the creation of first cell, first genetic material as in the form of RNA. Thereafter, there is encapsulation. And after encapsulation, there is a creation of cell. This is what the entity that created during timeline. Because of there is a vast difference between the polymerization reaction that is initiated by DNA polymerase and polymerization reaction initiated by RNA polymerase. In between that, there is a reverse transcriptase. The DNA polymerase that carried out the polymerization in long way. Because stringency of DNA polymerase is better as compared to RNA polymerase. That all this stringency level that always depends upon the ions that we called it as the chelating agents. These agents that plays very important role in order to stabilize the amino acids as well as the nucleic acids. There is a stability. Now the another thing is what the reverse transcriptase there come into picture. Single-stranded DNA to double-stranded DNA. This is what the transformation thing. But earlier, the material is the RNA as a genetic material. When we're talking about RNA as a genetic material, there are many more advances for the creation of RNA as a genetic material. Because of simplicity, need not require any 3' hydroxyl group for its polymerization, need not require a more number of accommodation of more number of nucleotides. The short stretch is able to do the job. Now these nucleotides able to drag the amino acid. It's a simple the affection process. And because of the affection, there is a gathering of amino acids. Because of the electromotive force, the joining of these amino acids, creation of polypeptide envelope. Only the thing is what? whatever encapsulation is there, that encapsulation that leads to the creation of only single entity. Now there is a joining of many more pieces of the RNA and the creation of huge biochemical entity inside the cytosol. Now creation of biomolecules, establishment of cytosol, get it biochemically activated, and ultimately there is a creation of cell. But why, one question is there, because RNA has a genetic material, that material is a beautiful material that created by nature. There is beautiful invention by the nature, need not require any intermittent molecule to carry out the job. So there is a creation of, or the incorporation of many more biochemical pathways by the virtue of creation of RNA. These biochemical pathways, they working efficiently to produce biomolecules, but still the problem is not resolved. Because whatever environment created in the Arctic is different than that of the Antarctica. But because of the tectonic plate shifting, earlier there is a cold desert. Due to the tectonic plate shifting, there is a shifting, and because of the shifting, the environmental condition is changing. Humid tropics to tropics, and tropics to temperate. During this traveling, during transportation, that cell face many more challenges. And because of the challenges, that cell needs to incorporate many more biochemical pathways. Now these biochemical pathways get it accommodated inside a small, tiny cell. Now there is urgent need to, because of the mixing of these all biochemical reaction inside the cytosol, that needs to be separated. Because in prokaryotes, only single cell entity is capable to do the job. Later on, there is the creation of mesosomes, the separation of respiratory reactions on the membrane, that is membrane bound reactions. Now earlier the life is heterotrophic, then gradually get it converted into chemiautotrophs, then to the autotrophs. Now heterotrophic life that consume much more nutrients, evolution of carbon, there is availability of sulfur. Then the heterotroph to chemiautotrophs, utilization of sulfur. After sulfur utilization, thereafter the consumption of carbon and liberation of oxygen. After liberation of oxygen, there is a creation of ozone, and ultimately there is a protection. During this transition, the mutations are there. Because of this mutation, there are भातकुलीच्या खेळामधली राजा आणि करानी अर्ध्यावरती डाव मोडला अधुरी what happens during this period? The creation of something new. Because of the mutation, then ultimately this trial and error. Because of the
WhatsApp
trial and error and
9767129963.
yeah. Please mute.
नागपूरचे सदरला
Because of the the situation, this is what madam, please. Mankar madam, please mute your mic. Yeah. Mankar madam. Mankar madam, please mute yourself now. Yeah.
Sorry sir.
Yeah. Okay. Now, because of the trial and error situation that created during that time. Now there is though there is a creation of this beautiful material as in the form of RNA, but this material, because this material capable to do the job very cleverly, did not require any intermediate to carry out the biochemical reaction. But still, nature wants to conserve information. For the sake of conservation of information, now there is a urgent need to convert this RNA into DNA. That too in the single stranded DNA. For that purpose, now there is a evolution of reverse transcriptase for the creation of single stranded DNA. Now the still the conservation is not there. Now there is urgent need to convert single stranded DNA to double stranded DNA. That's why the duplicate, the whatever information is there, that should be stored and duplicate because of the creation of double stranded DNA. Now you know the chromosomes and all other things that is in the two chromatids with two chromosomes with similar type of genetic material and duplication. Here is a duplication as in the form of strands, another duplication as in the form of chromosomes. Conversion of prokaryotes to eukaryotes. Compartment inside the cell. Formation of organelles. Separation of biochemical reaction. Distribution of power. This is what the scenario of evolution. Now there is a change from RNA to DNA. Because of the selective pressure, that creates the creation of deoxyribose first, then thymine, and proofreading mechanism that initiated by DNA polymerase. The fact behind the creation of or the conversion of RNA to DNA is very first is what deoxy containing phosphodiester backbone. Now this phosphodiester backbone is more stable in aqueous condition because each nucleotides that surround encircled by 4 water molecules. Because of the transitional metal ions. I already told you that the proofreading mechanism that not at all in RNA polymerase, but the proofreading mechanism that initiated in DNA. आता पुढे भातकुलीचा खेळ बंद होईल, right? The information in RNA that get it degraded because of the tendency of system to, there is a reductive deamination, reductively deaminated to uracil, the lack of correction, the conversion of uracil to cytosine revert back is not there. Fourth one is a UV eradication process. A large number of photochemical changes are there. In order to avoid the changes that created by the UV radiation, the photolyase is enzyme, is entity that rectify the changes inside the DNA. That's why there is urgent need to convert this RNA to DNA. But one thing is too much puzzling. Though the uracil is a stable base, uracil is a stable base. as compared to the thymine. Question is, in RNA, adenine, guanine, cytosine and uracil. These are the bases that construct RNA. On the other hand, adenine, guanine, cytosine that continue as a DNA base, but only there is a replacement of uracil that taken over by thymine. Why nature wants to continue adenine, guanine and cytosine as a base, DNA base? Why not uracil continue as a DNA base? Why uracil replaced by thymine? That's one thing. The second thing is what? Why nature wants to continue uracil as a base in RNAs till date, till today, all RNAs that continued by utilizing uracil. No replacement of uracil, right from back to 3.5 billion years ago. We continue. Uracil as a base. Here is there is a
तो ये आपको डिपेंड करता है कि आप हमारे कॉलेज को कैसा करते हैं कि वहाँ आपका पार्टिसिपेशन आपका प्रेजेंटेशन कैसा रहता है उस पर हमारे कॉलेज का अगला फेज
why and what sense the uracil continue as a RNA base? That not at all a preferred base for DNA? This is what a question. See, when we, because only small change is there. The conversion of or the replacement of thymine by uracil by thymine, replacement of ribose by deoxyribose is another change. When we consider chemically this thymine, the thymine is a uracil with one extra methyl group. Because, you know, when we're talking about DNA damage repair, the repair mechanism is a concrete mechanism that initiated by the DNA polymerases by the virtue of proofreading. Now, methyl group is a tag group. One extra methyl group that get it attached. That's why the there is urgent need to replace this uracil because RNA is a short-lived material. But DNA is a long-lived material that transfer from one generation to another generation. Whatever message is there, that message get it conveyed by the virtue of DNA. That's why there is urgent need of the replacement of uracil. Uracil replaced by thymine, but biochemically, the conversion of adenine to thymine is more favorable. Conversion of the cytosine to uracil is again favorable. This situation is favorable. No? Uracil not at all get it converted into thymine. Adenine to thymine conversion is more favorable thing. Because thymine have more potential because of the methyl group that initiated more proofreading potential, avoid the mutation, and ultimately there is a creation of DNA. Because of the DNA damage material repair mechanism, this cytosine is an entity that spontaneously turn into uracil. That's why uracil most preferably that utilize by the nature during primordial soup, during prebiotic environment for the creation of RNA as a material. Because you know, when there is the point mutation, uracil DNA glycosylase is an enzyme. This is a complex that evolved. And replace the uracil by cytosine because of the cytosine have to have more potential to get it converted into uracil. This is what the damage repair mechanism. That's why uracil is this. But another question is, why was the uracil written in the RNA? RNA is a short-lived, you know, I earlier told you the RNA is short. That need not require for long-term storage of genetic information. Cytosine that have to get it changed into the spontaneously into uracil. That uracil not at all a threat to the cell. Because why uracil retain inside the RNA is a big question. Because of the uracil, it's too much ambiguous molecule. The situation created by uracil is ambiguous situation. But still, inside the RNA, uracil is a beautiful molecule. The answer lies in the during the selection of genetic codes. Why uracil? Why not thymine? Because uracil is a beautiful molecule. But that molecule not at all get it continued. During the conversion of RNA to DNA. The answer to why uracil retain as it is. Because uracil is a too much ambiguous molecule. But still, uracil is ambiguous. Again, the strength of uracil is of the ambiguous nature of this uracil is a strength for the selection of codons. Now, you know, during the PCD, program cell death, uracil DNA glycosylase is enzyme. That enzyme capable to do the job. If too much ambiguous situation created by uracil. Now the signaling is of for the purpose of the program cell death, PCD. There are many more things. One of the most important thing lies with the uracil is what? The beneficial errors. Inside the vertebrate immune system, uricylated DNA is a because DNA did not have the uracil, but in the vertebrates in immune system, a hypermutation that start with deamination by the virtue of deaminase enzyme. Now there is a change of cytosine to uracil. That leads to that elicit an error-prone response for the sake of the proofreading mechanism. and repair responses. The differentiation of antibodies and all these things. That is because of the capacity to deaminate cytosine into uracil. That's why this molecule is a beautiful molecule for correction of errors. This is what the one thing. Second thing is what the ambiguous situation of this uracil. That's why nature wants to continue as a RNA base to maximal utilization of codes, genetic. That's why. But in order to avoid the rate of mutation, in order to avoid this ambiguous situation that created by uracil, there is urgent need to replace the uracil by thymine India. Let us move towards the code genomics. Because while talking about the uracil, why uracil needs to continue as a base in RNA. Sometimes they have to have more potential to DNA that go through the proofreading mechanism, that conversion of the reductive deamination of cytosine to uracil, and ultimately that create that mechanism. This is what one dynamic entity gifted to the uracil. You know the genetic code, but what happens? Because when there is a naturally there is a selection of the triploid code, but the combination is 64. Better way to keep 20 combinations, the single codon to each amino acid. But this is not the case. Code is triploid. When you consider the total 64 codons, then you will find the leucine that coded by 6 codons. Most of the amino acids that coded by 4 codons, like valine, serine, proline, throline, alanine, glycine, arginine. List of the amino acids that coded by the single codons. One more thing is what? One of the stop codon later on during evolution that get it converted into tryptophan. Because there is introduction of one more mechanism during evolution is of row dependent termination mechanism is earlier mechanism. Thereafter there is a introduction of row independent termination mechanism. At this instance, no need of the stop codons. That's why one of the stop codon get it converted into tryptophan. So if you consider the role of uracil and the genetic code, you will find cytosine and uracil, because there is a reactive deamination process that initiated by cytosine, get it converted into uracil. Uracil and cytosine at third position, Ubal base position, that always code for same amino acid. A and G open codes, same amino acid. C in middle, it's too much complicated. U in second place, code hydrophobic amino acid. See the role of uracil. Because of this ambiguous situation that created by uracil, codon-anticodon reaction that established. The establishment of codon-anticodon reaction. Now the uracil is pairing partner of adenine, pairing partner of thymine, pairing partner of guanine, pairing partner of cytosine, pairing partner of even uracil itself. So the specificity of codon to anticodon that generated by the Ubal, linking that generated by earlier two bases, first and middle. That's why uracil at last position. Because single amino acid coded by more than 4 codons. Somehow by 4, by 2. Still, there is a shifting of the ambiguous potential for the dragging of these amino acids. That's why uracil is important. Here is the anticodon preference rule. Second, third position. Guanine preferred first, then to the uracil.
हेलो पार्टिसिपेंट्स, आपको सर की आवाज क्लियर आ रही है क्या? बिकॉज फ्रॉम माय साइड इज ब्रेकिंग.
हाँ मैम आ रही
हाँ
है।
मैडम क्लियर है।
क्लियर नहीं आ रही।
Yes,
आ रही है।
madam.
बहुत ब्रेकिंग
मैम नहीं आ रही है मैम, शुरू से ही नहीं आ रही है।
क्योंकि
बहुत
मुझे
ब्रेक
वॉइस
हो रहा
ब्रेक
है
आ
आवाज।
रही है.
Yes,
हाँ
madam,
मैम
it is
बहुत ब्रेक हो रहा है, समझ भी नहीं आ रहा बहुत कुछ।
यस. प्रशांत सर, डॉक्टर प्रशांत सर. हेलो.
Yeah, madam.
अभी नहीं आ रही मैडम।
हाँ, कुछ कनेक्टिविटी का इशू है सर
हाँ
का.
मैडम.
सर आपकी वॉइस ना ब्रेक हो रही है. हेलो. हेलो सर, कैन यू हियर मी? हेलो. पार्टिसिपेंट्स, आपको मेरी आवाज आ रही है क्या?
हाँ मैम, आपकी आवाज आ रही है।
आ रही है मैम।
Yes, yes, I can hear you.
तो सर की आवाज बहुत देर से ब्रेक हो रही है।
हाँ हाँ, मुझे लगा कि मेरे तरफ से होगा, कोई पार्टिसिपेंट की तरफ से कुछ रिस्पांस नहीं दिखा, तो आई थॉट कि मेरे कनेक्शन में कुछ इश्यू है. हेलो प्रशांत सर.
ऑडिबल मैम.
देखते हैं सर का कुछ रिप्लाई आता है क्या. Just wait. हॅलो, डॉक्टर प्रशांत गावंडे सर, आवाज येतोय का? I think he lost his connection. ओके फ्रेंड्स, तो यहाँ पे हम रुकते हैं। सर की मीटिंग शायद लीव हो गई है एंड इट्स 12:36. हेलो, am I audible?
Yes, ma'am. Yes, ma'am.
Yes,
You are
ma'am.
audible.
Yeah.
Audible.
So
Yes, you are audible.
उनका शायद अभी टॉपिक बचा था कुछ एक्सप्लेन करने का, कंप्लीट नहीं हुआ है, बट अपना टाइम हो गया है लंच का एंड सर इज आउट ऑफ द मीटिंग. So I'll just call Dongarwar sir once and I'll let you know to whether stop the session or just a minute.
Hello, ma'am.
हाँ पार्टिसिपेंट्स, हम यहाँ पे रुकते हैं लंच ब्रेक के लिए. यू प्लीज प्रोसीड फॉर योर लंच ब्रेक, क्योंकि सर डोंगरवार सर का भी कॉल नहीं लग रहा है. तो वी विल मीट एट 1:30 फॉर द नेक्स्ट सेशन. थैंक यू ऑल ऑफ
Okay,
यू.
Meeting.
हाँ, मीटिंग लीव कर दीजिए क्योंकि सर भी अभी ज्वाइन नहीं कर पा रहे हैं. अपना लंच ब्रेक भी मिस हो जाएगा नहीं तो.
Excuse me.
अपन 1:30 बजे मिलते हैं. थैंक यू.
Thanks, ma'am.
थैंक यू.
Okay, ma'am.
Okay, ma'am. Thank you.
Thank you.